Now, we would like to present our experiment where we find out the blood group and blood type. The main purpose of conducting this experiment is to understand the basic concept of the ABO blood group system and to know our blood group and type. Materials required: toothpicks, blood sample, alcohol swabs, syringe needle, clean blood typing slide, sterile cotton balls, biohazard disposal container, and monoclonal antibodies anti-A, B, and D. Procedure: take a clean blood typing glass slide. Now wipe the finger with the alcohol swabs and rub gently near the fingertip where the blood sample will be collected. Prick the fingertip with the syringe and wipe off the first drop of the blood. As blood starts oozing out, allow it to fall on the 3 circles of the glass slide by gently pressing the fingertip. Apply pressure on the site where it was pricked, and to stop blood flow, use the cotton ball if required. Unpack the monoclonal antibodies kit. In the first circle, add anti-A. To the second circle, add anti-B. And to the third circle, add anti-D with the help of a dropper. Mix the blood sample gently with the help of a toothpick and wait for a minute to observe the result. Results: the tested blood is of group O and type positive, which is O positive. Precautions: discard the alcohol swabs, syringe, cotton balls, and toothpicks after their use. Drop all the materials, including the glass slide, into the biohazard disposal container after observing the results. Perform the experiment under the guidance of a lab technician. Sterilize the fingers properly before pricking the finger with needle. Use only new sterilized equipments. Perform the experiment under cool and sterile conditions. The surface of every red blood cell is coated with antigens, sugars and proteins that are integrally linked to membrane proteins or lipids. The clinical relevance of these antigens for blood component transfusion and tissue or organ transplantation lies in the ability of these surface molecules to incite an immune response, potentially causing a transfusion reaction or organ or tissue rejection. In addition, some red blood cell surface antigens have cellular functions with clinical relevance, and others are targets of immune attack in certain infections. The membrane of each red blood cell contains millions of antigens that are ignored by the immune system. However, when patients receive blood transfusions, their immune systems will attack any donor red blood cell that contain antigens that differ from their self-antigens. Therefore, ensuring that the antigens of transfused red blood cells match those of the patient's red blood cell is essential for a safe blood transfusion. Red blood cell antigens can be sugars or proteins. Blood group antigens are either sugars or proteins, and they are attached to various components in the red blood cell membrane. For example, the antigens of the ABO blood group are sugar. They are produced by a series of reactions in which enzymes catalyze the transfer of sugar units. A person's DNA determines the type of enzymes they have, and therefore the type of sugar antigens that end up on their red blood cells. In contrast, the antigens of the Rh blood group are proteins. A person's DNA holds the information for producing the protein antigens. The Rhd gene encodes the D antigen, which is a large protein on the red blood cell membrane. Some people have a version of the gene that does not produce D antigen, and therefore the Rhd protein is absent from their red blood cells. People with type A blood group have the antigen A on the surface of their red blood cells, while people with type B blood group have the B antigen. Individuals with type AB blood have both A and B antigens, whereas people with type O blood have no antigens present. The antibody that reacts against antigen A is called anti-A and is found in the plasma of people who do not possess antigen A on their red blood cells. That is, people with type O or type B blood. The antibody that reacts against antigen B is called anti-B and is present in those who do not possess antigen B in their red blood cells, people with type O or type E.